genetics are involved in preventing diseases like hereditary versus lifestyle, diet, these things play into
there's a bunch of ways of answering it. We often talk about gene and environment.
So, Judge, want to hear the question?
No.
Yep. Okay. She was just wondering, sorry, I just wanted to repeat for everyone, and she was wondering how important is genetics versus lifestyle in terms of...
They're both important. We usually use the word gene or environment. Environment means it's not the environment like the weather necessarily. It's environment meaning the variables or factors that you can't control theoretically. You know, if you drive a lot, you're more likely to get into an accident. So that's the environmental risk. People who smoke are more likely to get lung cancer. So most lung cancer, much lung cancer is associated with smoking. So for each of the cancers, there's no one answer to your question. It depends on, again, on the cancer. What I try to do, and I explain it to our students in terms of genetic contribution, is every cancer has a small fraction, meaning 2 or 3 people out of 100 who have a strong predisposition based on a strong gene that's running in their family, or it's what's called a new mutation when they were conceived at birth, just bad luck, or passing down through the family, like hereditary breast cancer. So every cancer, that's leukemia, lymphoma, colon, breast, brain, lung, all the pancreas, all have these small fractions of cases. Those are very interesting scientifically. And sometimes if you find the cause of those rare cases or infrequent cases, it helps you understand the more common cases that are called sporadic. In terms of the genetic contribution to sporadic, that's hot debate in the past 5 years, because we now have ways to look at genetic variation in any individual for about $500. So we can take your blood cells, which are the easiest ones to get, or cells from your inside of your mouth, buccal cells they're called, get the DNA out of them, and then run them on what's called a chip, and we can look at the variation in you. We can look at 500,000 or 1 million sequences of DNA and look at yours compared to the person beside you and see where that variation is. And that variation explains a lot of things, you know, your hair color, your height and weight, and everything, and many other differences between you and the person beside you. But that also probably explains what's going to happen to you, your fate, and that's determined at conception. And those variations are, sometimes they're called SNPs, or single nucleotide polymorphisms. Now, any individual variation is not going to make a big difference. What we think is happening is, I told you about here, is the accumulation of multiple different abnormalities is what predicts your fate. And this is what people are trying to find out now, is patterns, variations in your DNA that predicts your future. And there's companies in the U.S. that are making a lot of money where you can send in your DNA and they'll say you're 4 times likely to get asthma or heart disease or colon cancer. And there's some truth to that, but you have to be careful interpreting this data, because the environment, the other factors that you talked about, are very important as well. So that's kind of the long-winded answer. So I think both contribute. Again, there's no one answer for one cancer for you. If you ask me what your chances are of getting asthma, the asthma scientists probably give you a very sophisticated answer based on genetics and environment.
There's an interesting observation in my field that could sort of lead to that question as well. And again, a lot of times when we're answering questions, I'm afraid sometimes we're not really giving you clear answers because we're introducing more complexity, because there may not be a direct answer yet. But there is an example of a paper that was published by a really well-known stem cell scientist called Sarah Kenner 2 years ago, where he had this rare opportunity that scientists never get, to study a pair of monozygotic twins, identical twins, who were born, 2 girls, were born to a couple, and one of the girls got leukemia when she was 2. And they studied her blood. They took cells from her blood, and they studied some of her stem cell populations. Because I think, in terms of cancer stem cells, the one cancer where it's really well established that we think it comes from a stem cell is probably leukemia. And so they've actually identified this fusion gene, an AML fusion gene, that when you have that fusion gene, it gives rise to leukemia. It generates leukemia. So they found that the twin sister who got leukemia, obviously she had that fusion gene, and we have a pair of every gene on each chromosome. So both of her genes were fusion genes, and therefore she expressed the disease. But the really interesting thing is when they took the blood from her sister, the unaffected sister, the healthy sister who did not have any leukemia, one of her parent genes had the AML fusion. And yet she was completely healthy. So what could that tell us? That may tell us something about the interaction of genes and environment. First of all, from our perspective, the genes were only mutated in the stem cell population. And here's a child who's completely healthy with a stem cell population that harbors one mutation. And so the question is, what should we do with this child? Is there some environmental exposure that she's going to get that we don't know? Because we don't know all of the environmental exposures that cause cancer. That's going to trigger a mutation in the other allele, and maybe is she going to get leukemia later in her life? And why was her sister unlucky to get that environmental exposure and get that other gene, assuming she wasn't born with it? What caused her to get that exposure? And so it brings up actually more questions about the fact that both are important, I think. You are born with genes that predispose you to getting cancer, with mutations that predispose you to getting cancer. And then on top of that, if you already have a weakness and you have one hit, one gene, all it's going to take is some environmental exposure to hit the other, and then you might get the expression of cancer. So it's going to be really interesting to see a follow-up study on whether this other twin maybe remains healthy all her life, or whether she might get cancer much later. It's very interesting.
Would that be the same as with HIV? Carrier, but you don't necessarily get the disease?
Yeah, that's a good analogy. So the analogy was that what if you could be a carrier of a disease but not have that disease? And that's a very good point. So that's, for example, like cystic fibrosis or other diseases where we know that there's a genetic mutation that accounts for that disease. That's exactly what happens. You have people who are carriers and they don't know they're carriers. And so technically, could any of us be carriers of cancer? And what environmental exposure could be causing the expression of the cancer? It also explains why the older you get, the more likely you are to get cancer. Right? Steve talked about how 30% of the population is going to get cancer. Well, there's an age predisposition to that, and luckily for my small patients, it's very rare to get cancer as a child. It's much more common to get cancer when you're 80 or 90, because your cells are aging and accumulating all those mutations. So it's a probability thing. It becomes more likely as your whole genome degenerates, acquires mutations. So.
Yes, I guess adding on to the story about the twins, I was just curious if there are ongoing current research underway on the area of epigenetics, and if so, what area you could elaborate on it. And particularly, I'm interested to see if there's anything being done on understanding the role of stress on the stem cells and the genes, etc. As we know, with having high levels of cortisol suppresses the immune system, and so what the connection of that may be if there's anything in that area.
That's an excellent question too, and I think that there is no doubt that there's a lot of evidence in all kinds of different fields that stress in many different forms could theoretically weaken the immune system and perhaps weaken the surveillance such that you may have expression of disease. And there's all kinds of stress and disease links. So for example, with cardiovascular disease, it's a much stronger link. Stress is actually coming close to one of the risk factors now for cardiovascular disease. But with cancer, I think stress is still not on that list of risk factors. Not because it's not important, only because we haven't succeeded yet to figure out what are the multifactorial causes of that stress and what are the biological substrates of that stress. For example, could stress lead to a decrease in your DNA repair capacity? Could stress knock out some enzymes that are really important to repair your DNA? And is that why when you're stressed you get cancer? We don't know the answer to those questions yet. But I suspect as people study more the biological substrates of stress, there will be answers. And the second part of the question, epigenetics, is there's lots and lots of really good research going on in epigenetics in every field of cancer now. And particularly in leukemia, I don't know if I know But yeah, in leukemia, there's a lot of epigenetic discoveries now that have been made that contribute to leukemia. And in particular, for my field, brain cancer, one of the most prominent drugs now that's out there to treat glioblastoma is called temozolomide. And it's a chemo agent that's been around for a long time. And actually, someone brought up before, like how to pick agents. We don't actually, a lot of these chemo agents, we don't even know their mechanisms. A lot of them are derived from plants and they've been used for a long time, and some of them are based on alkaloids or salicylic acid even, and a lot of them come from just, they're very non-specific, but they work, and that's why we use them. So anyways, in terms of epigenetics, temozolomide is an alkylating agent that's been around for a long time, but it really increases survival in a certain percentage of patients with glioblastoma. And the reason is because it has an epigenetic mechanism that was discovered in 2004. And actually, in patients who have silencing at a certain DNA repair gene called MGMT, those patients respond really well to temozolomide. Because the temozolomide can come and knock down all the cancer cells, and the cancer cells have lost their ability to repair themselves, so they just die. So in those patients who have that gene silenced, which is an epigenetic, a specific epigenetic mechanism, then those people respond really well to this drug. So that's kind of a backwards way of learning about the drug, that we discovered something in retrospect. And now we know there's a certain population of patients that respond to that drug.
Okay, we're coming close to 8 o'clock, so we have time for one more question from the audience.
I just have a question about personalized medicine, personalized therapy. There are lots of talks about like these days. And the question is, if there are these stem cells or cancer-originating cells, and they are constantly mutating, is it possible for it to develop any personalized therapy? Because I do a situation like this flu, anti-flu vaccine, to make this vaccine, and then next year it mutates, and we have to start it all over again. So what's the view on this personalized therapy?
Yeah, you guys are a pretty smart crowd, and that's a great question. And so this has been addressed in various cancer stem cell symposiums around the world, under talks of really interesting titles. So people have called cancer stem cells moving target. And then some people will say cancer stem cells, are they cells or are they a state of being, right? The point is that, you know, if we want to actually say what is a cancer stem cell, to some degree you have to invoke a Heisenberg uncertainty principle, because we really don't know if a cell is ever the same cell when it's in the same place at the same time. And so as a result, it really is a moving target. So your question is excellent. How do we hit a moving target? How do we, by the time we personalize our medicine and discover a very targeted therapy that will hit one patient's cancer stem cell population, what's to say that that population hasn't evolved, right? And so I think we're going to learn by trial and error as we move some of our targeted specific therapies into patients. We're going to be able to figure out what's the success rate of these targeted therapies, and that will tell us how quickly these populations are evolving. It'll follow the same path as retrovirals, right? It'll follow the same path as HIV therapy, where how quickly do you have to evolve your drug to catch up with the changing virus. So it'll be a similar story, but I think for a lot of solid cancers, we're not there yet with therapies in the patients. With leukemia, they're getting there very, very shortly.
Okay, I know it's so tough to do this one. There are so many fantastic questions and comments from the audience, but just cognizant of the time, and I do want to finish up with one last question that I'll address to all of the panelists. Maybe it's in advance, so we can see what their answer is like. I thought I'd ask, based on our topic today, new approaches to treating cancer, which of these new approaches do you think will have the biggest effect, the biggest impact on the discovery of effectively a cure for the various kinds of cancers?
Well, I think it's a bit of a loaded question, because I think the three fields we talked about all represent targeted chemotherapy, even though I don't think absolutely do that. I think they're in different stages of research and development. So targeted chemotherapy is here, and there are a lot of concrete examples. So you'll hear about them in the next few years. I think there's a lot of activity and interest, so it's working. I think stem cells And cell therapy is very, well, cell therapy also has a practical application, but I think they're more of the R&D phase. So, in 10 years, we don't know what's going to happen, and I don't think they're certainly not competing. They're complementary. So, I think you'll see. I think it's really more related to the timing of development of these